our speaker today, Dr. Dallas Donohoe, who comes from the University of Tennessee, Knoxville, where he's a professor of nutrition there. So just to give you a little overview of his history, he graduated from Florida State University, a fellow Seminole, and then he did his PhD at Louisiana State University and his postdoc at the University of Chapel Hill, or University of North Carolina in Chapel Hill. So if you could give a warm welcome today, he's going to present his presentation, Targeting Cellular Metabolism to Understand the Anti-Cancer Actions of the Fiber Metabolite Butyrate. So I think it's going to be a very exciting seminar. If you could give him a nice, hearty welcome here today.
So I'm going to start the talk by giving a little bit of a background, and we're going to learn a little bit about the microbiome and fiber, and then we'll get into butyrate. And so this is actually a transmission electron micrograph from a C57 black 6 mouse. It's showing the colonic lumen and the colonocytes along here, and you can see the brush border. And when I did this experiment, originally we're supposed to flush the colon. I forgot to flush the colon. But they turned out to be really awesome images, as you can see, because they really illustrate the symbiotic relationship that we have with these microbes and how really close they are to us. And so this has led into what we call the microbiome. So each and every one of us are colonized by microbes that exist on and within us. Most of these are bacteria. They can also be archaea and eukarya. And the majority of these microbes live within our gastrointestinal tract. It should be noted that this relationship is absolutely essential for survival. Okay, there's been many years where we've tried to rear these germ-free mice or animals that have no microbiota and have been unsuccessful. And that's because these microbiota are required. This is another image, and this kind of goes to the second point, and that is that these microbiota provide both genetic and biological diversity. So here you can see each one of these colonocytes, and you see the vast number of these microbes. So it's estimated that they outnumber us based on cell number by 10-fold, okay, meaning that you're actually 90% bacteria and only 10% human. In addition, they also provide genetic diversity. Their genomes, collectively termed the microbiome, outnumbers us in terms of protein coding genes, our own human genome, by 100-fold. And so they're doing a lot. This has led to what's called the Human Microbiome Roadmap Initiative Project by the NIH. And this has used a lot of sequencing, metagenomic sequencing, to really identify who's there. Now, I believe at the beginning of this, they probably thought it would be more like the Caribbean, clear waters, where we'd all have, disease states would have a single microbiome, and we could define, and we could use that to basically identify these disease states. But as it turns out, even normal individuals, everybody has a different microbiome. So it's more complicated. It's turned into the Tennessee mud bath, you might say. And so it defines who's there. In addition, we're also left, now that the Human Microbiome Project is completed, and we pretty much know who's there in terms of bacteria, there's a need for more functional studies. And so what is the impact on host health and disease? Some of these come about through studying the role of bacterial metabolites in health and disease. This shows just some of the diseases and disorders that have been implicated in terms of or regulated by the microbiome. The one I'm going to talk to you today about is colorectal cancer. And so my journey, I kind of think of it, sometimes I'm on the yellow brick road. I'm progressing. And I think the microbiome project started when I was in UNC. So I went from LSU to UNC. And now I'm at the University of Tennessee. And eventually, a couple of years from now, it'll all blow up when I go for tenure. So this is kind of the way I see things. When I was at University of North Carolina, I was very lucky, okay? I happened to jump on a project at the time that was hot and it was interesting, and people were beginning to use notobiotics, or not beginning to, they were actually being reacquainted with using notobiotics. And I joined a lab where we were using Metabotics and we're using germ-free mice. And the original thing that I did was I actually took germ-free mice, and these are mice that are raised in these isolators in a Metabotic facility, so they're sterile. And I basically compared those to conventionally raised mice. And I did a metabolic analysis. But the reason I did that metabolic analysis, I told you I did that, those initial transmission electron micrograph experiments. And these TEM experiments kind of really sparked my interest, okay? So here we see, this is an SPF, all right? You can kind of see this little bacteria hanging out here. And you see the you can see the brush border, you see the colonocytes, you can see the bacteria, the mitochondria here, okay, in the colonocyte. When I looked at germ-free, number one, they had a ton of these little black dots, which are called matrix granules. And I didn't know what to think of that, okay? And at first we thought it was a staining artifact, but as we did each mouse, it kept showing up, and it never would show up in SPF. You always had this abundance of these black dots. And it turns out in the literature that they're representative of mitochondrial dysfunction, all right? And so this kind of let us know that there's perhaps a mitochondrial dysfunction or something metabolically going on. Second, that was very interesting was in the germ-free, we found a lot of these, what would be double membrane vesicles or autophagosomes, okay? So this also showed us that something was going on, and this also can happen in an energy-deprived state. In addition, one last thing that I don't know how to follow up on, but I always think about in the back of my mind, even the position of the mitochondria was different in the germ-free. If you notice here, the mitochondria, in all cases, they almost came up to the brush border. Here they seem to be pushed back. And so these are just observational skills, which are very important in science. But it led us to kind of ask the question, if we just took out these different tissues and we just did two energy biomarkers, let's say like the NADH-NAD ratio and ATP, would we see differences in a germ-free and an SPF mouse? And we didn't see differences in pretty much all the tissues we tested, with the exception of the colon. The colon was exclusively affected, where the NADH-NAD ratio was decreased and the ATP level was also decreased. And so this suggests that the microbiome has a strong contribution to regulating energy metabolism in colonic tissue. At the same time, we took and isolated colonocytes from germ-free mice and SPF mice, and we did transcriptome profiling. And we ran all of the genes that were affected through a KEGG analysis, and we also did proteomics, mass spec, and we also ran those through a KEGG analysis. And if you look at the top pathways, butyrate metabolism came up as number 1 and number 2 in our proteome, okay? And so this suggests that germ-free colonocytes are in a starved state due to the absence of perhaps butyrate. So what is butyrate? So butyrate comes from fiber. Fiber is basically the non-digestible component of our diet. Fiber escapes absorption and digestion in the small intestine. It makes it to the colon, where we have specific bacteria that can ferment fiber into many different metabolites and peptides, or metabolites. 3 of these main ones are short-chain fatty acids, which include acetate, propionate, and butyrate. All of these are found in millimolar concentrations. Butyrate is unique in 2 things. Number 1, it's the primary energy source of the colonocyte. This was done back in the 1980s by Rodinger, where he took C14 labeled butyrate and C14 labeled glucose and looked at the preferential usage of both of those molecules and found that butyrate was preferentially used by the colonocyte. Second, in the 70s, butyrate was also identified as the first inhibitor of a class of enzymes which are now known as lysine deacetylases, histone deacetylases, okay? These molecules basically, or these proteins remove acetyl groups from histones and other proteins too. And so butyrate is a HDAC inhibitor and an energy source. And so I'm going to summarize from the germ-free SPF paper. We found that microbiota exert strong effects on energy metabolism in the colon. This is partially due to lack of butyrate, because we could rescue these effects just by adding back butyrate. And it was not due to HDAC, because we used other HDAC inhibitors that did not rescue. It seemed to be primarily its role as an energy source. And so at the same time, it's always good as a postdoc or a graduate student to multitask. If something fails, you have something to fall back on. And so in this case, I was lucky it succeeded, but I also was doing this project. And as from a pharmacology background, I really asked the question of how is butyrate an HDAC inhibitor, considering the fact, I didn't mention this yet, it's a very weak HDAC inhibitor. So the Ki has been reported anywhere between 80 and 300 micromolar, and so it seemed not very likely that it could ever accumulate to that level to inhibit HDACs to begin with. And secondly, how is this related to its usage as an energy source? And third, what's this different in a non-cancer versus a cancerous colonocyte? And the reason I say that is because although the colonocytes primarily use butyrate, as they turn to a cancerous colonocyte, glucose utilization increases, okay? And so in that, they undergo a process called the Warburg effect. And in this case, they can take up to 10 to 100 times more glucose. They increase glucose utilization through aerobic glycolysis. And a lot of this is because glucose is just a great building block, okay? And it can be used to build DNTPs, lipids, amino acids, and NADPH, which are all necessary components for a proliferating cell. And we know that this happens because clinically, through using PET imaging and using radiolabeled glucose, we use that as a way of basically identifying, we can be used as a way of identifying colorectal cancer and many other types of cancers. And so our hypothesis was that the Warburg effect changes the fate of butyrate and regulates its action as an HDAC inhibitor. In a normal colonocyte, it's mainly utilized and burned as CO2 through beta oxidation. And in a cancerous colonocyte, it's taken up, although in that case, glucose is utilized and it might have some sparing effect. Okay, very simple. We also wanted to see whether the Warburg effect could modulate some of butyrate's actions on cancer cells. In this case, these are HEC116 cells, which are colorectal cancer cells. And what we found was that low concentrations, when we would inhibit the Warburg effect, either through taking away glucose or through knocking down different proteins such as LDHA, we found that butyrate, not only did it not decrease proliferation, but it actually had promoted proliferation, okay? And so this is very interesting. However, as you got higher in concentration, you'd see the same sort of decrease there. When we looked at just global histone acetylation, so this is H3 acetylation, as you would expect, butyrate with the Warburg effect, and this is normal, you have a dose-dependent increase in histone acetylation. However, when we knocked out the Warburg effect, we found that at lower concentrations, butyrate can no longer increase histone acetylation significantly, okay? And when we measured C13 butyrate in the cell, we found that this is 24 hours pulse experiment using C14 labeled. We found that 5 millimolar, it could reach concentrations of up to 400 micromolar, whereas at 0.5, the highest concentration, even with the Warburg effect, was 40 micromolar. So this brought about another question. That is, if it's so low, how is it increasing histone acetylation at lower concentrations? That was the basic question. So then we started to basically propose where butyrate could act as an HDAC inhibitor. This most likely at high concentrations, where at low concentrations, it's oxidized, goes to acetyl-CoA to citrate. The citrate shuttle transports citrate back out to citrate. Citrate then is converted to acetyl-CoA by an enzyme called ATP citrate lyase. This acetyl-CoA is a necessary cofactor for histone acetyltransferases. So I mentioned that histone deacetylases remove acetyl groups. Histone acetyltransferases add acetyl groups. And so to test this, what we did was we proposed to just knock down ACL and look and see how butyrate would affect histone acetylation at first. And so in addition, when we would inhibit the Warburg effect, one of the things that kind of led us to ACL was we'd see that it was also decreased significantly. And so This shows you a nice dose-dependent increase. However, at lower doses, we could never get an increase with butyrate at 0.5 millimolar when we knock down ACL. And so this suggested that it was working through this pathway, but really we wanted a confirmation experiment. There's always that experiment that confirms things. And so what we did was we took C13 labeled butyrate. All 4 carbons were labeled with C13, and we wanted to see how much C13 acetate, or the 2 carbons would be C13, would come out if we isolated histones, okay? We hydrolyzed off those acetyl groups and then measured that by LC mass spec, okay? So, and these are pulsed for up to 3 days. And basically, first, this shows you, number 1, we can get incorporation of basically these labeled carbons into these acetyl groups. And if we knock down ACL, we decrease that, okay? Showing that this mechanism is indeed relevant. And so what's happening is you have at lower concentrations in normal cells, you have an ACL-dependent mechanism, okay, through HATs. And at higher doses, we believe it's through HDACs. To further test that and to kind of get at some of the targets, what we did was we wanted to use this RNA interference technology, where we would knock out or knock down ACL. And so normally butyrate would upregulate gene A. Okay, when we knock down ACL, if it no longer regulated increased gene A, we would say that that gene was ACL dependent. Okay, and the same way, if it upregulated gene A and we knocked down ACL and it still upregulated gene A, then we would say that it was ACL independent. And so what we found was this, where at lower doses at 0.5 millimolar, 75% of the genes, when we knocked down ACL, they went away, okay? 25% of the genes did not. So 75% were ACL dependent, 25% were independent. And we assumed that it was HDAC, but we don't know, okay? As you increase dose, it went from 75 to 42 being dependent. And at 5, you had a complete reversal where 75% of the genes were HDAC independent and only 25% were dependent. And when we took these genes and we ran them through a KEGG analysis software, we found that a lot of the ACL dependent genes were basically regulating cell cycle, and ACL independent seemed to be correlate with cell death. And so this paper kind of showed that the Warburg effect results in butyrate having opposite effects on proliferation. So at lower doses, it can promote in the absence of the Warburg effect. Metabolism plays a role. Less butyrate metabolized equals more accumulating and elevated HDAC activity. Butyrate at low, medium doses regulates histone acetylation as acetyl-CoA donor. And so at the same time, we were starting to get in a gnotobiotic isolator, and we wanted to look how important butyrate is and a high-fiber diet is in terms of regulating colorectal tumorigenesis. Dietary fiber and its protection of colorectal cancer is actually controversial. So as many papers that show protections, there's just as many papers that show that it doesn't protect. We wanted to basically, but these are all human epidemiological studies, so we wanted to control the microbiome and see if we could see a protective effect, and whether these protective effects of a high-fiber diet and, let's say, butyrate were dependent on the microbes. And so what we did was we had 3 different isolators, and we used a BALB-C strain and an AOM DSS to induce our colorectal tumors. And each of the isolators, you can't give DSS to a germ-free mouse, you'll kill it. They're immunocompromised. So we had to create a situation where we had a defined microbiota, but we didn't want any butyrate producers. So there are Alter-Schlater flora, ASF strains that are very well defined. We chose 4 of these based on their ability to not produce butyrate. And so we colonized these isolators, these mice, with these 4 ASF strains, and we put them on a low and high fiber diet. In addition, we also had another isolator where we had the same, we colonized the same ASF, and we put on a low fiber diet, but we wanted to see if you didn't have a butyrate producing bacteria, if we added a butyrate fortified diet, could we still see a colorectal tumor prevention or decrease the multiplicity or incidence? And so our third condition, we had the same 4 ASF strains, plus we added in this butyrate fiber solvents on a low fiber and high fiber diet. This shows you the data here. I'm always very afraid when I do animal studies because I'm afraid I will end up with nothing at the end. So I'm kind of gun shy when it comes to those experiments. So this is probably one of the most nervous days in my whole postdoctoral experience, but it turned out to be great and very, very awakening. So this shows you the low fiber minus VFib. High fiber had no effect in terms of preventing. However, when we added the VFib, we went from 9 tumors to 3 tumors, and they were smaller, and the grade, they also were at a lower grade based on histopathology. In addition, when we added tributyrin, we had even a greater effect at 2. And this is just an example, of a low fiber diet. And when I opened up these tributyrin, a lot of these mice had no tumors at all, okay? And so this is very interesting. So fiber protects against tumorigenesis in a microbiota-dependent way. So you see all these human epidemiological studies that show no protective effect, right? Well, what if we classified those patients or those people in terms of microbiome status? Butyrate producers, non-butyrate producers, because that's different among everybody. Tributyrin protects against tumorigenesis even in the absence of a butyrate producer. Butyrate accumulates, and I didn't show you this data, but we actually found that it can accumulate in colorectal tumors, both in mouse and human tumors.
So tributyrate is a prodrug for butyrate, right?
Yes. I'm sorry. Yes. So tributyrin is basically just like a triglyceride. You have 3 fatty acids linked to a backbone glycerol. This is basically butyrate linked to a backbone glycerol, 3 butyrates. So starting a faculty position at University of Tennessee, it's very important in any case to make yourself unique. At the University of Tennessee, I did not have a gnotobiotic facility. And so I had to take my research in a different direction, number one, from my postdoctoral mentor, but define myself. And so I wanted to address not necessarily the microbiome, but what butyrate was doing to the colonocyte and the cancerous colonocyte in terms of metabolism. And I wanted to use an instrument called the seahorse to measure butyrate oxidation and see what was regulating that. And so the seahorse is an instrument shown here where we can grow cells on these plates, and we can look at the oxygen consumption rate, and we can look at the extracellular acidification rate, okay? And this is a relative proxy of lactate. Going back to some of the original, things that kind of pushed this, this is a paper from the Blanchard lab, and this is using C14 labeled butyrate and acetate, and look at the oxidation in these colorectal cancer cell lines. Here you can see that acetate, you keep going up, whereas butyrate, somewhere between 1 and 2 millimolar, you actually top off, all right? This is kind of something that sticks in the back of my mind all the time, and it suggests that there's a capacity in terms of the oxidation of butyrate. So to use the seahorse to measure butyrate oxidation, it's currently used to measure long-chain fatty acid oxidation, and the basis of that is long-chain fatty acids, basically, with the long-chain fatty acid CoA, the CPT1 exchanges the carnitine for the CoA, and you have the long-chain fatty acid carnitine, which then could be taken up into the mitochondria. It's important to realize this is the rate-limiting step in beta oxidation. It's a very important step, but the basis of measuring fatty acid oxidation with this instrument is we use a fatty acid oxidation meter that just has a very low glucose, 2.5 millimolar glucose, and then we inject in etomoxar, which blocks CPT1 and uptake of that long-chain fatty acid. And that gives us a proxy of the fatty acid oxidation. Unfortunately, for short-chain fatty acids, I should say, they're believed to not use this carnitine-dependent mechanism, right? And so we really can't use etomoxar to block short-chain fatty acid oxidation. So I had to devise a way to measure that. So instead of knocking out the fatty acid oxidation, I decided to knock out glucose oxidation. So normally the media has glucose, and it's measuring oxygen consumption rate, and the oxygen consumption rate is due to glucose utilization. Now when you inject in butyrate, you have both of those substrates, and then you inject in 2DG, that leaves you only with butyrate contributing to the oxygen consumption rate. And so that's showing the area under the curve there. So this is one of the initial experiments where once I inject butyrate, now they have glucose and butyrate. You can see there's no difference there. Now when you add 2DG, you see that there's a nice dose response going from 0.5, 1, 2, and 5. Okay. And so once I developed that, now we used it to compare different cell lines. So we used the HEC116 cell line, compared it to the fetal human colonocyte. So this is not a cancer cell line. And we found that the fetal human colonocyte had elevated butyrate oxidation and elevated oxidation in general, as shown here. So it suggested that the cancerous colonocyte had a decreased capacity to oxidize butyrate. When we measured certain metabolites like carnitine, what we found in the FHC cells, and I should say the last experiment was done at 5 millimolar butyrate. So when we measured certain metabolites like carnitine, we found that in HEC116 cells, it was very low. And we looked at the expression of the transporter, OCTN2, it was much higher in FHC cells compared to HEC116 cells. So this suggested that carnitine might play a role. And just to review, once again, carnitine is necessary for the uptake of long-chain fatty acids. It's not believed to be up for short-chain fatty acids. Nevertheless, when we take carnitine out of our media, what we found was that the butyrate oxidation was significantly reduced. Okay, and this is 5 millimolar concentration of butyrate. However, when we use 1 millimolar or lower dose, we found that taking away carnitine had no effect on butyrate oxidation. So this was only at higher doses. Don't believe everything you read in the literature. And so in the science books, it tells you that butyrate's carnitine independent. And for the most part, that's true. However, in the colon, we're dealing with millimolar concentrations. And a lot of the earlier experiments, they're comparing basically palmitate, which is made up in micromolar, to butyrate in micromolar. And even then, they find a 1.6-fold increase. But as you go up in concentration, things change. In addition, different cell types were used. This is showing you, as you increase the carnitine, your butyrate oxidation goes up at 5 millimolar. And so we wanted to begin to test this with basically using pharmacology with etomoxar and also knocking down CPT1A. And so knocking down CPT1A, we get a nice dose response, and we show that it's functionally relevant because we basically eliminate palmitate oxidation. In addition, when we test it with 5 millimolar butyrate, we find that compared to SI-MOC, where we got a nice increase in butyrate oxidation, when we knock down CPT1A, we know butyrate is no longer oxidized. And because of this, we find that if you're oxidizing it, our assumption was that it would accumulate and you could actually, or if it wasn't being oxidized, it would accumulate and HDAC activity would be affected. And we find that at 5 millimolar, HDAC activity is higher in the carnitine compared to the non-carnitine, and acetyl H3 follows along that, where the acetyl H3 is higher in the minus carnitine, and the plus carnitine is decreased in both the 1 and the 5. Now, the reason we didn't see any difference in the HDAC activity for the 1 millimolar, and we did for the histone acetylation, what we think is because these experiments were done between 3 and 6 hours. This experiment was done between 24 hours. Okay, so building a model. Don't get too caught up on it. Basically, butyrate goes through a CPT1 independent and dependent pathway. And what regulates this? What's regulating carnitine? And going back to the Warburg effect in cancer cells. And so we were very interested in whether if we block the Warburg effect or we slowed it down, how would that affect both carnitine transport and the carnitine transport OCTN2 and carnitine levels. And so what we did was we used an inhibitor called DCA, which inhibits pyruvate dehydrogenase kinase. And so normally in cancer cells, PDH is phosphorylated. This inactivates it, all right? And so we wanted to activate it, so reverse the Warburg effect in some way. And so when we add DCA, we were able to decrease PDH, but we also noticed that OCTN2 went down and OCTN2 went up and CPT1 went back up. So this is suggesting that the Warburg effect is suppressing OCTN2 and CPT1A. And interestingly, what we found the most interesting thing was that when we used DCA, carnitine we no longer could see this carnitine-dependent and independent butyrate oxidation. So it seemed like the Warburg effect was a major regulator of that. And so butyrate oxidation in the cancerous colonocytes is CP2-1 dependent and independent at higher concentrations. Elevated glucose utilization decreases carnitine uptake and CPT1A. This results in diminished butyrate oxidation at higher doses. So with that, I'm going to conclude my talk. Work was done by Anna Hahn, a graduate student in my lab, and Megan Johnstone, who, another graduate student. And it was also helped whenever you're at an undergraduate institution, undergraduates play a big role in a lot of the science that goes on. So Natalie Bennett was a big driver. And I also like to thank a lot of my postdoctoral mentors, Scott Boltman at University of North Carolina. Jay Whalen was a collaborator, and Sears Bioscience. With that, I'll take questions. Yes.
I really enjoy your lecture. I think it's very appropriate for this class. As you summarize every section, I think that's great for the students. Really
Thank you.
capture the information. I was just wondering, it's fascinating to see this butyrate effect on the mitochondria, energetic. If anybody have studied butyrate on mitochondria, like brown mitochondrial function in brown adipocytes?
Yes. Actually, I'm collaborating with somebody that does not brown adipocyte, or not brown. I know white adipocytes. I'm familiar with. It tends to. I have to get this right. You read so many papers. So, in terms of the adipocyte, it tends to increase energy utilization. It doesn't cause mitochondrial dysfunction in the adipocyte per se. It's been used. Is that what you're asking in terms of?
Yeah, because mitochondria content in brown adipocyte is much higher than
Yes,
white
yes,
adipocyte,
yes.
which is that is really that the function instead of generate ATP, they generate heat.
Mm-hmm.
So I think that would be interesting to see that. Also, you talked about these 2DG blocking glucose.
Yes.
That would be interesting to investigate. And so I thought you might
Yeah.
have knowledge.
Yeah. So I'm actually there is a new faculty member who's doing brown adipocytes. We haven't done butyrate oxidation yet with the seahorse, but that's something that we've talked about. So. But in terms of mitochondrial dysfunction or mitochondrial function, it's burned pretty quickly. So I'd imagine that they would burn it really fast. And we could do a mitochondrial stress test and look at things like uncoupling and see how it affects uncoupling and parameters like that and see. But I'm not familiar with anything out there right now.
One summary slide after the low and high fiber diets.
Mm-hmm.
I missed the last point. Butyrate accumulates
in tumors. So so we actually measured butyrate in colorectal tumors from humans, and we actually found that it was higher than the adjacent normal tissue. And we also, from our mouse studies, we also had those tumors, and we found that it was higher than the normal tissue. And that was normalized to protein. So that was that last point.
I had a comment and a question. I thought the observation you made at the beginning about the relative position of mitochondria in colonocytes, the ultraviolet light was very interesting. It reminded me of some studies I was aware of 5 or 6 years ago in marathon runners and long distance runners in muscle. They developed different mitochondria populations closer or lower down in the cells. And they have very different energetics. So it'd be interesting to follow up on.
Yeah.
I had a question about OCTN2. At the end, you showed a chemical effect on the expression of OCTN2. Did you use genetic methods to knock down or overexpress?
No, we're doing those experiments now. So yeah. That they actually have knockout mice too. We want to follow up on this story and use those. This was just a down and dirty pharmacological DCA, and so I should say, at least treatment with DCA is associated with decreasing OCD2, but we'd have to do the knockdown to kind of do a better confirmation of that. So I know it's a lot of data, a little data heavy maybe. So someday when you guys give talks, you'll learn, oh, maybe I shouldn't have given so much data. Maybe it's definitely a give and take.